This is what I was saying about everywhere being so quiet during the lockdown. It's like a reminiscent of what happened in March, basically. Hi guys! Welcome to my fourth PhD vlog. Today is 15th of January 2021. So this is my first vlog of the year and it's currently 8:30ish. Again, about to grab breakfast and then go to the lab. And I'm sorry if my energy is sort of low today. Actually, it was really hard for me to pick the camera today because it's one of those days where I've had a very long week. So today is Friday, right? And I feel really tired. The way I'm treating YouTube is like my work. I find it hard to do things halfway. Anytime I decide I want to do something, I always end up putting my whole heart at it. So it's either I don't do it at all or do it to the best that I can. And this is something that it can be good, but it can also be bad. It could be good in the sense that you try to strive for excellence in everything that you're doing. But the bad side is that sometimes you just don't start things because you know that you would not be able to do it to the best way that you would have imagined for yourself. Am I the only one that struggles with this? Please let me know in the comments if you are like that. My primary work is my PhD, of course, but I'm treating YouTube like a full-time work, and if I'm going to treat YouTube that way, that means that I aspire to release video every week, at least once a week, because consistency is very important for me. So with that mindset, that's why I decided that I have to pick up the camera today, because today is the last day of the week. And also I mentioned that this journey has its lows and its highs, although it's uncomfortable to watch someone in low energy, I guess. My intention for this channel was to try to be as real as I can. So at the moment there's a national lockdown in the UK and it started at the very beginning of the year, so everyone is indoors. We're not allowed to go out except from some certain exceptions. Whenever I go to the lab or video myself around, I do have it in mind that it's such a privilege to be able to do all of these things, because I know that a great majority of people globally aren't able to continue with their education or able to continue their work because of this pandemic issue. So yeah, I just wanted to put that out there. As I mentioned earlier, there was a pre-Christmas deadline that I set for myself in my last vlog, but unfortunately I wasn't able to meet that deadline. So I don't know how people handle not being able to meet your expectations. That's how I felt like. I felt like I was slacking behind. I'm trying to delineate the things that I can control and the things that I can't control. And I tried my best to sort of make sure I meet that deadline. But just because of logistics and how the world works, I couldn't. This week, I was trying to finally get that deadline over with. And also just hearing about the COVID cases, and all of it is having a toll on my mental health, sort of. I'm also battling with insomnia, as I mentioned from my previous vlog. I'm not saying this to dump all of my emotional problems onto you, but I'm saying this just to give you context as to why I might not be my usual upbeat self, because there are lots of things that I'm just trying to sort of figure out, and I always try my best to remain positive, optimistic, and making these videos for you all is also a good way for me to feel like I'm doing something meaningful, something impactful despite all the things that are happening globally. By making these videos, I hope that it's somewhat able to transpire some sort of positive energy to you. After when I eat, I would take the bus and go to the lab. So I'll see you once I get to the lab. Toodles! So it turns out that I missed my bus. That's it. I don't know if you can see it, but it's so annoying because now I have to wait for 15 minutes in the cold. What a perfect way to start the day. I don't know if you can see it, but that's it. And it's still waiting. But the thing is that I can't run, because if I run, there's no way I can catch it. So it's so painful to miss it and then to see it pass right in front of you. So I'm just going to wait till it passes, just so you can have a rough idea of how painful it feels to me at the moment. That's it. That is it. Great. This is what I was saying about everywhere being so quiet during the lockdown. It's like a reminiscent of what happened in March, basically. I'm usually the only one in the bus. Like, it's almost like I have my own personal driver. Everywhere is super quiet and you hardly find cars moving. Look at really strange times, to be very honest. Like, although yeah, the quietness is a bit peaceful, but you know, this kind of quietness is peaceful but at the same time it's disturbing. That's exactly how it feels. Hi guys, so I arrived not too long ago. It's currently 10:01. Who says 10:01? It's currently 10:01, and yeah, it's super weird because I woke up like since I think 6:30 or so, and I'm just getting to the lab around 10. Super weird, but anyways, so now I'm going to write down my to-do list as usual, and yeah, I'll take it from there, I guess. This is not in a particular order, but I think the primary thing for me today is to pull and send my samples for sequencing, and I also want to send an email about pathological assessment at some point next week of my samples. I also want to send TB request and fill in the sequencing form before sending this for sequencing. Actually, also continue with my literature review, which I'll talk about later on, and then there's a lab meeting as usual at 3 p.m. So in order to explain what I've been up to, I had to write like the illustration of my workflow basically, and I'm going to show you the workflow, and that will hopefully explain what I've been doing up until now and why today is very important for me. So when I get human tissue, whether it's a normal tissue or a cancerous tissue, what I do is that I microdissect the tissue, which I've vlogged about at some point. After microdissecting, I extract DNA. When I finish extracting the DNA, I prepare the DNA for sequencing, and then when I finish preparing the DNA for sequencing, I finally sequence it and then analyze the data. So since November, I've been working from here up until here, and then I was preparing for DNA sequencing for about since maybe mid-December. I want to send it for sequencing today. So this is the workflow, and basically I've spent about 3 months working on this, more or less. Today is the day that finally all of the experiments that I started since November are going to end, before I then continue with the data analysis, which will take forever as well. So for I don't know how basic I need to go explanation wise, but for those who don't know, so the DNA is basically a very long molecule. I'll leave a picture here. It's probably something that you guys are familiar with, the 2 strands that looks like a zipper. That's your DNA basically, and that's a long strand of molecule that contains genetic information that is needed for a cell to function. And this long molecule is basically made up of 4 chemical compounds, which are 4 letters called bases. I don't want to use terminologies here, so I'll just call them 4 letters. And so you can imagine that DNA is a very long stretch of sentence that contains just 4 letters, and depending on how those 4 letters are arranged or sequenced, that would tell us the kind of gene that is expressed in that DNA. So what sequencing does is that it looks at the orders in which these letters are arranged in the DNA that we're sequencing. So when I say I'm sequencing, that's what I mean. I want to check the order of the letters along the stretch of the DNA, and by checking the order of letters, I'm able to know the genetic, if there's a change in genetic expression or if there's a change in gene at that area. And you can imagine that in a disease like cancer, you would imagine that the long stretch of DNA would be different. So the idea for sequencing is that we know the normal sequence, the normal gene, how the letters, 4 letters are arranged. But in the case of cancer, we're trying to figure out what are the changes that we see. Changes that we see, are they the ones driving the cancer? So when I say sequencing, it's very, very complex. My best way of explaining this in a simplistic way that anyone can understand, when I say sequencing, that's essentially what it means. So we're trying to find the orders of letters here, and from the orders of letters we're able to know, okay, Is there something wrong? Could it be because of this cancer? So that's what I'm trying to do. So as I mentioned earlier, I've extracted DNA from some of the samples that I'm looking at, and now I'm going to send it for sequencing. And the thing with sequencing is that it's very expensive. That's another fun fact. So we want to make sure that what you're sending in for sequencing is good quality samples. And as I mentioned, that I've been working on these samples for almost 3 months now. So I'm hoping that like everything goes well and the results look like good quality data, because at the end it's the data that would speak for itself. So all of the effort that you put in, no one sees that. It's the kind of data that you generate that would speak for you. Sequencing isn't done here, so there's the sequencing facility that is like 10 minutes away from where I'm working with about 54 to 55 samples, give or take. So what I want to do today is that to pull all of the samples, because if you're sending for sequencing, you can't send them individually, you have to pull them together. So what I'm doing today is that I want to pull all of the samples, and after pulling them, fill in the sequencing sheet form. After filling that, then submit my samples at the sequencing facility. That's how today is going to work, and that's what sequencing is. I hope it sort of makes a bit of sense for anyone, and I'm sorry if this is basic for some people. I just don't know the kind of like audience that I have. If you guys are a bit familiar with biological stuff or not, so I just decided to do it basic. Like, it's better to be too basic than too complex and speaking gibberish. Hi guys, so Jeff and I finished talking about the pulling and how it's going to happen. I showed him the calculations that I made and all of that, just so we were on the same page. At the moment the time is 12:58, and I hope to be done with all of this before 3 p.m. because that's when the lab meetings start. I'm panting because I just climbed the staircase from going to pray, so in case you noticed, that's why. So now I'm just going to go back to like my experiment and stuff, and then I'll catch you guys later. I finished pulling the first cohort with about 25 samples, so now I'm going to move into the second cohort with about 29 samples. Labeling of tubes is super important because that's like the foundation of all of your experiment and everything that you want to do. And I spend like a good amount of time just labeling and making sure that like the numbers are properly written and I can read it properly. I don't know if you can see this, but yeah, just make sure that the numbers are obvious, because if you mess up from here, everything else they're going to transfer or pipette would just be shambles. Like, you can make sense of your data if you label wrongly. Quite a mundane thing to do, to be very honest, but it does help you on the long run if you like very meticulous about this step. So I spent about an hour and a half or so pulling all my samples, and then when you pull your samples, you have an expected volume that you're supposed to get when you're pipetting. So when I pulled my samples and then I checked the volume of my final sample, I found out that it's not exactly accurate. So I had like a few microliters less than I expected, and that can only mean one thing. It's either pipetting error or there's something wrong with the pipette's calibration, because I tried to be super careful when I was pipetting. So I would say my pipette has not been calibrated recently, so I guess that's why I didn't get the required volume. So now I just have to hope for the best that the differences in volume doesn't make a huge impact on the data that we get from the sequencing facility. So just added his own sample to my pool, and he's gone upstairs for the quality check that I mentioned about. I'll share the data with you once we get it. So after the quality check, we're going to send it off to the sequencing facility. So now I have to pray through first, and then after praying through, I have to fill in the form that I talked about earlier, and it's 1 minute to the lab meeting, so I have to pray through, rush back upstairs real quick such that I can tune into the lab meeting as I fill in the form. So I'll catch you guys soon. Hi guys, so I've just finished with praying and I've tuned it to the lab meeting now. So now just to tell you what I'm doing, I'm trying to fill in the sequencing form now, finally. And while I wait for Sujas to come back with the quality control data. Hi guys, so it's currently 5:07. In the past hour or so, I have sort of been filling the sequencing form, and finally Sujas ran the samples via the quality control that I told you about earlier, and samples look quite okay. I would walk you through the results later on when I'm rounding up, but we finally sent it to the sequencing facility. I'm not the one doing it actually, so Sujas kindly agreed to help me drop the samples off. So he has gone to drop samples off now for sequencing, and now what I would do is that I want to respond to an email or a couple of emails, and then I would have to clear my bench. Yeah, I'm quite tired, so I'm sorry if I come across as I have low energy. I haven't eaten anything all day, so my energy levels just dropped, but otherwise I'll go to writing now. So I was just thinking about the to-do list that I wrote earlier on when I first got here, and did everything except from the literature review. And I sort of actually, I think, predicted that I wouldn't be able to do it, but I was hoping to be optimistic to squeeze in some time during the day, just like maybe an hour on it, since my deadline is in a couple of weeks. But I wasn't able to do that, so hopefully I can focus on that during the weekend. We'll see about that. So now that I've tidied up a bit, I can show you. It looks decent. Not the perfect bench, but I think I did quite a good job clearing everything up. So I said that I was going to show you my data from the quality control. I'm not sure how clear this is, but this is how the quality control looks like. So this is the first cohort and the second cohort. So this cohort has over around 36 samples, whereas this cohort has around 24 samples. And in terms of, so you can see that there's a nice band. I don't know if you can remember from my first vlog where I didn't see anything like it was like this for my first vlog, the time that I was trying to optimize. So that is a sign that you don't have DNA in your sample, so you don't have anything in it. But you can see that there's a band here, so this band means that I have my DNA here, I have my DNA here ready for sequencing. So basically that's how the data looks like, the quality control. So Sujat and I concluded that the libraries are okay. I'm really hoping that when I get the data, it looks good. So I guess the question now is that what's next? Am I done with my experiments? So can I travel now and just relax? No, unfortunately no. Even though I've sent it for sequencing, there are tons of samples that I still have to start from scratch, extracting DNA, making libraries and all of that. From the sequencing facility, it takes almost a week or 2 weeks sometimes to sort of get your result back. sometimes it could take a month. So there was a time that I actually sent in my first year. It took me, I think, a month and a half or 2 months before I got my result back from the sequencing facility. So when I was talking earlier, my battery died. It takes a while before I get my result back from the sequencing facility, so it wouldn't be advisable for me to just be waiting. So I have to start other experiments. So now what I'm going to do is I first have to recalibrate my pipettes, because I need to minimize the kind of pipette or errors that I have, because as from the result from today shows that there's something wrong with my pipettes. Next week basically I'm going to get new cases from new patients, microdissect them, extract the DNA, make the libraries, and finally send them for sequence. So it's going to be like a loop, a cycle where I go back and back until I get the cohort size that I want for this study. I think that's pretty much it for me. Again, I'm so sorry if I'm looking like I'm speaking with lower energy and my facial expression is like dull. I'm so sorry for that. Today is one of those days where I'm just really tired, and I felt really overwhelmed at some point today because I had lots of things happening, but I'll be fine. I just need to have a proper rest. Hopefully I hope to sleep, have a good rest during the weekend, feel recharged such that I can pick up from where I left on Monday. I think I'm going to round the vlog off, so thank you so much if you stuck around with me to the end. I'm super grateful. Please help me grow my channel by clicking the like button and also subscribing if you haven't already. If you're feeling super kind or generous, you can also share my videos with your family and your friends, and leave a comment as well if you like. If there's something you found interesting all through the vlog, mention it in the comment section. I'm always excited to hear from you all in the comment section, so it doesn't just feel like I'm talking to myself. I hope to see you on my next one. Bye.